All right, business as usual. If you're watching this video to get any credit for it, you're going to write down every time I say an attendance word throughout this lecture. I also take a screenshot every time I say one of those attendance words. And then at the end of the video, send an email that includes the list of words and screenshots, and also one question about the material covered in this video. So for you watching the video, the first word for attendance today is lab. So write down lab, and I should see a screenshot of me pointing at the word lab. And that's it. Now, it is 7:30, so you can skip ahead about 30 minutes into the video, and that's when the lecture will start. Morning.
Morning.
Okay. Let's see how much time we have here. Not enough. All right. Morning.
Morning.
All right, showing. Ashley is here. All right. Barrett's not here yet. Wooten, not here. Trahan, not here yet. Dickens?
Here.
Dickens here, right. Clark? Not here yet. Inca's not here yet. Zaya's here. Smith is not here yet. Watson is not here yet. Kaylee is not here yet. Lucas is here. Marley is not here yet. Ballard, not here yet. Savannah is here. Mia, not here yet. Edwards, right? Yep. Edwards is here. Wilkes is not here yet. Barrett came in while I started. Did I miss anybody? Inca's here. We have less than a minute, so I'll start some of the redundant reminders and stuff. Even though it's not written there, as always, remind you to check your emails before you come in to make sure we're actually meeting. Obviously, we're meeting today. Here we are. One thing that's also not written up there is, I had some issues, so it took me a while to finally record the Monday's lecture. So I recorded it, I just haven't posted it. I'll post that today. It's kind of weird, right? Because technically I started chapter 3, and now we're gonna jump in the middle of it. You guys haven't seen chapter 3, one of you have, as the rest of you haven't, but it's okay. The information in chapter 3, it's not important, because probably not gonna be tested on it. And it's not cumulative. So even though you didn't learn the stuff I taught when I recorded the lecture, you don't need to know that to understand the stuff I'm gonna teach you today. Does that make sense? Okay. Anyway, let's see, who came in? Last name? Wilkes, that's right, gotcha. I think I have everybody, it's 8, so. Schaefer, right?
Yeah.
All right. So anyway, yeah, that's that. So some good news today. We don't have a pre-lab on Friday. So, every week you have a pre-lab due, there's not one due this Friday. Because the next lab we're gonna do is so important, we're breaking it up into 2 weeks. So next week, all we're gonna do is meet. We're gonna come in here and meet for lab. I'm gonna give you a whole pre-lab lecture to really go into detail about how that lab's gonna work and how to write a lab report. And then next week, after you have that pre-lab lecture, then your pre-lab will be due next Friday. And then the week after that is when we'll do the actual lab itself. Smith, right? All right. Smith is here. So any questions about pre-labs for this week? They don't exist. All right, Smith is here. Yeah, exam 1. Man, we've been so crazy with the schedule, weather and all that stuff. Things have to keep getting pushed back. We're a little bit behind. Me. Your hand, right? Okay. Got it. Okay. Anyway, yeah, because things have been pushed back, like I said, at first I was planning for us to do this exam in person so I could proctor it, but we just don't have time. So it's open. If it's not open now, it will be when we're done with lab. But it's going to close on Sunday. So you have now until then to do it whenever you want. You have 50 minutes. So whenever you found the time to take it, you can take it. Does that make sense? All right. But everything else is still the same. It still covers chapter 1, 2, and 3. And I say 3 technically because there's probably hardly going to be any questions from chapter 3, which is what we're about to cover today. There's not going to be any questions about that on the exam. Your study guides are your quizzes, quizzes 1 and quizzes 2. You have about 50 minutes to answer 50 questions. It's closed book. What else? Oh yeah, when you're done with it, you're all going to be able to retake it. So a little bit less pressure. So what will happen is after it closes on Sunday, on Monday it'll be opened up and you can retake it as many times as you want. And the retake is not timed and the retake is open book. And once you're done retaking it, you're satisfied with your new grade, you're going to email me and I'll average your first grade and your highest grade. Does that make sense? So it's nice and easy, less pressure. If somehow some weird thing happens and you can't get a higher grade on your retake, even though it's untimed and open book, we'll just keep your original grade. If you don't email me to tell me your grade needs to be changed, it won't change. So there's also no pressure. Also the final exam, half of the final exam is going to be based on the first 3 exams. So exam 1 will be your study guide for the final exam. So even if you get a good grade, it would still help you to retake that exam 1, even if it's just for practice. What else to say about that? And just like any other assignment, once it's closed and it's been graded, I can meet you online and give you feedback. In this case, give you all the correct answers to the exam, which you're going to need to study for exam 1. And just like any other thing in the semester, these assignments, if I meet you and give you feedback, I give you a 5% boost to your grade. So I'm hoping this really eliminates some pressure for you. Right? The exams are important. You're going to be able to retake it. You'll be able to get a 5% boost and all that good stuff. So any questions about the exam? All right, I mentioned this on the last lecture, but I haven't posted it yet, so you haven't seen it. I just want to say, if you're feeling overwhelmed, don't. This is a really weird semester. At the beginning of any semester is always the hardest. Because not only are you learning the material, but you're learning how the course works, like when, where do we meet, and how do I find the assignments, and how do I submit the assignments. It's a lot. And then after that, the fact that we barely met, we've barely been with each other because of the weather. So if you're feeling like you're overwhelmed or you're behind, you might be. But let me help you. It's okay. Don't feel bad. I'm being a little bit lenient on things like due dates and stuff like that, and I'm definitely here to help you get caught up. So if you feel like you need help getting caught up, let me know. I'll definitely help you with that. And this is only exam 1, right? So this means we're a quarter of the way through the semester. So you got plenty of time to turn things around. So anyway, any questions about that? All right. As usual, attendance is same thing. So you're watching the video, I've already told you what to do. I don't think there's anybody live streaming. But for those of you who are here, you're going to send me an email at the end. But this time, I am going to ask you to write down the attendance words and then ask one question about the material covered. That makes sense. All right. So let's jump into it. Let's see here. So the first word for attendance is going to be exam. So you can write that down if you're here, because at the end of this, you're going to send me an email sometime before 9 a.m. It has a list of words, and the first word will be exam. All right. So let's jump into this. And again, I feel really bad that I couldn't get this posted in time, because you didn't see the first part of chapter 3. And one of the things I really reiterated when I started this chapter was, as far as academics are concerned, this is a very unimportant chapter. Because it's hard, like I said, there's not going to be any questions for the most part about this on the exam. And also, the stuff you're going to learn in the first half of the semester is all about cells, right? So you're going to learn how cells do photosynthesis. You're going to learn how cells do respiration. You're going to learn how cells make protein. You're going to learn how cells reproduce, all that stuff. You're going to learn all that in other chapters, right? After this chapter, you're going to learn all that stuff. So that's when you're really going to learn all the important parts of the plant of the cells. So again, chapter 3, because I teach out of the textbook, I'm giving it to you out of a formality, sort of, because again, it's a textbook, so I'm presenting it to you. But yeah, I'm going to go through it quickly for this reason, because again, academically, this is unimportant. We'll learn the important stuff later. So let's jump into it. So last thing I was starting to talk about that you guys haven't seen yet is I'll talk about the difference between plant and animal cells, comparing and contrasting. Right now, I'm kind of doing the comparing and talking about how they're similar. So there's these things called intracellular junctions. You do not need to know about these. That's about putting a big X to it. But again, I'm teaching out of the textbook. So really quickly, I'm going to introduce you to the concepts, then we'll move forward and not talk about it probably ever again in the semester. But anyway, these intracellular junctions, it is what it sounds like. It's these junctions between cells. This is where they have direct contact. They use this for communication. There's a word called plasmodesmata, and that is the junction between plant cells. And again, you don't need to know that. I'm just presenting this because it's from the textbook. Animal cells also include contacts, but those are called tight and gap junctions, but they also have desmosomes. But again, I'm only presenting this because it's in the textbook. I'm not going to ask you anything about that. But do you have any questions about that? Okay, the next slide is just pictures. Again, very unimportant. You can see we have 4 different pictures. We can see a plasmodesmata right here. This shows this connection between the 2 cells. These are 2 separate cells, but you can see they're connected now because of this, and they can also, share stuff, right? They can share molecules, cell water, whatever it may be, communicate with each other. Here we have what we call a tight junction between 2 cells. Yeah, and I'm only explaining this because some people, I just realized we have to be ADA compliant. I didn't think about that. So when I give these videos, I need to give an audio description of the visuals. So anyway, that's that. And then here, you can see some more cells together. All these parts, right? You can see all these parts that are named and labeled. Completely unimportant for us. I'm not going to ask you any of this. I'm putting a big old X through here. But again, I just wanted to quickly at least introduce you to these terms to say that we did learn chapter 3. So any questions about that? Okay, well, this is good because this next portion of chapter 3, which is 3.4, we're going to talk about the cell membrane. And this actually is a little bit more important for things that you're going to learn later in the semester. But again, for exam 1, which is what's due on Sunday, technically it covers chapter 3. But even this, all the stuff I'm about to teach you is important for later, but not for exam 1. By the end of the section, you should be able to understand the fluid mosaic model of membranes and describe the functions of phospholipids, proteins, and carbohydrates in membranes. And again, for exam 1, this is unimportant. First of all, we'll talk about the fluid mosaic model. This describes the structure of the plasma membrane as a mosaic of fluid components, which basically means, and again, you guys haven't seen the first lecture, but when I first started talking about cells, the first thing I talked about was the cell membrane, which is what we see in this picture. It's that phospholipid bilayer. The cell membrane is what keeps everything in, right? Without the cell membrane, you wouldn't have a cell. That's what contains the cell. It also allows things to come in and out, which is what these things are, right? You can see these little proteins that we'll talk about later. So again, this is just an introduction. That'll be important later. But yeah, and this picture that we see here, again, it's a phospholipid bilayer. It's like we've cut into a cell membrane and we're looking at it. like we've sliced into it, we've looked at it like that. You can see these things called glycoproteins, which I'm not going to teach you about because for us, they don't matter for the rest of the semester. None of this stuff. Now, this will be important later. Again, we do have these protein channels that will allow things in and out. Think about what we're doing in lab today, right? We're doing a diffusion and osmosis experiment where we have like a fake cell membrane and we're going to see what kind of stuff comes in and out of it. Same here. Of course, those things don't have protein channels, but same concept. Any questions about this? All right. So, animal cells have cholesterols in the membrane. Plant cells do not. And this is to regulate fluidity. It's basically like an antifreeze. And again, I'm not going to ask you any of this on the exam, so I'll put it next to this. This is just to introduce you to the concepts. Do you have any questions about that slide? All right. The membrane is mostly a phospholipid bilayer. I already mentioned that in the last lecture that you guys haven't got to see yet. But yeah, it's mostly phospholipid bilayer. As far as we're concerned, if you ever were to get a test question on that later in the semester, we just consider it a phospholipid bilayer, right? That's the main portion of it. But there are the other things that we talked about with that last picture, right? The proteins, the glycoproteins, all the other stuff that'll be important later, but it's not important yet. I'm not going to test you on this, but this is important to keep in the back of your mind. The surface, I'm going to back up. The surface is hydrophilic. Where did you at? There we go. Hydrophilic, and the inside is hydrophobic. Because if you don't know if you remember, when we talked about in chapter 2, we were talking about these biological molecules, one of which was lipids. And then we talked about different types of lipids. One of those was called a phospholipid, and that's what each one of these little things is a phospholipid. So you've got a little phosphate group there, and then you have the fatty acids, 2 fatty acids. And as you remember, and as you do need to know for the exam, so throw back to chapter 2, remember fatty acids and lipids are hydrophobic as far as we're concerned. But again, when we're talking about the cell membrane, that's a slight exception because the outside is indeed hydrophilic, which will matter because that kind of matters later in the semester when we talk about things coming in and out of the cell. That's part of what dictates what can come in and out of the cell. How big is it? Is it hydrophobic? Is it hydrophilic? Is it charged? Is it polar? All those things that we'll talk about later in the semester. So again, the major component of the cell membrane is those phospholipid bilayers. The second major component is the proteins itself. And again, you saw that picture that has little blue proteins embedded in the membrane. They have a lot of jobs. They work as channels or pumps, again, controlling and regulating what comes in and out of cells, which will be important later in the semester. They also have enzymes, and you've been introduced to enzymes. We haven't talked too much about them yet. After this exam, we're really going to start talking about enzymes a lot more. Basically, as you should know, the thing you should know about enzymes is they're proteins. That part's from chapter 2, so you do need to know that. Mostly proteins. There are some exceptions, but we're not concerned with those exceptions in this class. But yeah, enzymes are basically proteins that make chemical reactions happen. So almost all the chemical reactions that happen inside your cell don't happen without those enzymes. So they're important for that reason. And again, that'll be an important concept later in the semester. This is also the attachment for the cytoskeleton. Cytoskeleton was something I talked about on the last lecture that you guys haven't seen yet. But as far as we're concerned, again, academically, the cytoskeleton is not important. You don't need to understand that concept to understand things that we're going to learn later in the semester. And then finally, these proteins also help with cell recognition. So for example, like the COVID vaccine, it's an mRNA vaccine, and all that mRNA does, it provides an instruction on how to build a particular protein, just the protein that's on the outside of the virus itself. So that protein that's on the outside doesn't hurt you, but it does help with cell recognition, and that helps your immune system be able to fight it. So again, for this exam, exam 1, none of this is important. Later in the semester, these 2 will be. Channels, right? Again, regulating what comes in and out of the cell, like we're going to cover today, but also these enzymes that make chemical reactions happen. Any questions about this slide? All right, next word for attendance, I'll point to it. It is model, M-O-D-E-L. Model is the next word for attendance. So again, after this, sometime before 9 a.m., send me an email with that list of words and 1 question based on the material covered in today's lecture. All right. So that's the second major component. Third major component, carbohydrates. So there's carbohydrates embedded in the cell membrane. These are always found on the outside. So when we talk about those proteins, as I mentioned previously, sometimes they're on the outside, sometimes they're on the inside, sometimes they're large enough where they kind of bridge the gap between both. But here, they're always on the outside. They're always bound to lipids or proteins, so it's never just a carbohydrate on its own. And again, this is used for cell recognition. As you can see, there's a lot of ways the cells recognize each other. Really quickly, I put a big old X through this. It's a visual reminder. This will not be on the exam. And as a matter of fact, this will never come back in the semester. So again, academically, this is very unimportant information. You do not need to understand this to understand the things you're going to learn later in the semester. But it's in the textbook, so here we are. Any questions about that? Okay, here's where there's an exception, so I'm going to start slowing down. These next two portions, passive transport and active transport, this will be more important a little bit for this exam, but also very much for learning things later in the semester. So a lot of the stuff I've covered, including the stuff you haven't seen yet, like I said, will be important later in the semester because we're going to cover it later in the semester. But in this case, it will be important later semester, and now is when we're covering it. Does that make sense? So if you've been nodding off, now's the time to pay attention. Let's talk about passive transport. So as we've already mentioned before, membranes are selectively permeable, which means some things can come in and out, some things can't. We talked about that earlier when I was talking about how the outside is hydrophilic and the inside is hydrophobic, right? There's all kinds of things. Sometimes things are too large to just willy-nilly diffuse in and out of the membrane. But yeah, obviously this is important because your cells need to control their environment, right? When you learned in chapter 1, one of those properties of life was the ability to regulate your environment, right? This is part of how they do that. It's part of how cells do that. They keep concentrations, certain concentrations of molecules within their cells. So yeah, any questions so far? And really quickly to describe what you see on screen, other than the words that I just talked about, what we have here is a picture, again, a cross section, like we're looking at a cell membrane. There's that phospholipid bilayer, there's a phospholipid bilayer. This is a protein that basically just acts like a doorway or a hallway, if you want to call it that. So whatever these little balls are, they cannot make it through the membrane for whatever reason. Maybe it's a size thing, maybe it's a chemistry thing, doesn't matter. They can't make it through. So instead, it goes through basically this little hallway, this little protein just lets it come through. And we'll talk more about that later today. And on this side of the image, it's sort of the same thing. We have these little balls that cannot go through the membrane, so they need some assistance. In this case, it's a protein. You can see it's like changes shape. What that's indicating, that we'll talk about later, is that that protein requires energy. So instead of it just kind of slide through it like it is on the left, this one goes into it, it changes shape, and kind of spits it out the other end. And again, we'll cover that later today. But are there any questions so far? Okay, if you download the PowerPoint lecture, you can click this little video to see some of this stuff in action, animated. Same with this. This one's a little bit more realistic. Again, you can download the PowerPoint, watch this little video. It's just an animation showing you things being transported through the membrane. All right, this is good stuff. This is a really good time. I'm going to pretend like I did this on purpose. I keep saying we're behind, but no, I did this on purpose. Since we're going to do this in lab today, now you're learning about it, right? So let's talk about it. Diffusion, right? That's basically, well, almost entirely what this lab's going to be about. Diffusion and osmosis. When you think about it, and you'll learn later, osmosis is basically like a very specific version of a type of diffusion. But anyway, what is diffusion? There's so many different definitions, as I'm sure you guys have noticed from doing the pre-lab. But one of the definitions is it's the movement of molecules from an area of high concentration to an area of low concentration until equilibrium is reached. And this does not require an input of energy. For example, if I were to light a scented candle right now, right over here, first, you guys in the front 2 rows would smell it. Then you guys in the back would smell it. But at first, it would be like a very heavy scent here. And the farther away you moved, it would be less noticeable. But eventually, all those molecules would spread around throughout the room. And no matter where you were, it would be the same scent. That's diffusion. And again, it doesn't require input of energy. I don't need to sit here and fan it to get those molecules to go out to you. Now, could do that, but I wouldn't need to. They would diffuse. So what we're going to talk about is, as an example on the next slide, we're going to imagine a membrane that's separating pure water from a mixture of dye dissolved in water. So again, we're just talking about diffusion. Before I get to that side, I like these pictures. These are not from your textbook. And another thing I don't think that's in your textbook that is in your lab that you're hopefully familiar with already is this idea of Brownian motion. Does anybody remember hopefully what Brownian motion is? That's when these molecules basically move, right? Molecules move, they don't sit still. And I love this picture, even though you don't need to know anything about it, but it's a great example of how water moves. And in this case, this is what we're talking about, right? We have these things in water. So in this case, hopefully this looks familiar to you. You've drawn a water molecule before. You have this one oxygen and the hydrogen. And you can see what they're trying to show here is this water molecule just basically stretches. Like imagine that thing going like this. And that's exactly what that's doing, is stretching. This one's doing the same thing, but at different times, right? It's moving like this. This thing's moving like this, almost like it's flapping its wings, if you want to call it that. Right? This thing is rotating. Rotating that way. This one is rotating this way. This one's rotating that way, right? So basically this is, again, just one great example of what molecules are doing. They're not just sitting still. And remember when we talked about temperature, when you add temperature to something, when things are getting hotter, what we're saying here is there's more molecular motion. So this is what they're doing. So again, just imagine these things sitting next to each other. They're going to hit each other and knock off and go, flying off somewhere else, and then hit other molecules and hit the glass. In this case, we have them in a beaker. So that's how they move around, right? They're basically banging into each other. So here we go. Here's a picture that I was talking about. I think this is from the old textbook. This is a great example of diffusion. What we have here on the left is when we start, right? So we have this thing of water with this semi-permeable membrane in the middle. That's what that yellow thing is. And it lets these red things through, right? It's whatever it is, the red things have the right size and chemistry to get through that membrane. So what you cannot see, because this is a picture and not a video, is these little things are just bouncing around. Just try to imagine them bouncing around, and again, randomly, they're going to make it through those holes. And eventually, it's going to be to the point where they have what we call equilibrium, where you have an equal concentration on both sides. Just because that's just how nature works, right? These things are just going to bounce around off of each other until there's equal on both sides. That's important to note, not academically, because I'm not going to get this detailed on exams, but when we reach equilibrium, I do love this picture because you can see this arrow, for example, indicates that ball is going to be going that way, right? This arrow indicates that ball is going to be going that way. This arrow indicates that ball is still moving around, it's just staying on that one side. The point is, when equilibrium is reached, there's no more net movement. Meaning, yes, some balls, in this case balls, these little red balls, yes, they will be moving back and forth, but there is no net movement, meaning concentration on this side is generally going to stay the same as the concentration on that side. Despite the fact they're going back and forth, they're going back and forth at an equal rate. So any questions about that side? Next slide is basically the same thing, except now we're throwing in a different color dye, right? So again, we have this semi-permeable membrane. And on the very left, this is the initial picture. So we've got these blue balls bouncing around, and you can't see the fact that they're bouncing around because it's an image, not a video, but they are. Same with the red balls. And eventually, they start moving back and forth, which is what this middle picture is. And then finally, just like on the last one on the right-hand side, we have equilibrium, right? So we have an equal concentration of red, 1, 2, 3 balls on the right, 1, 2, 3 balls on the left, equal concentration of the blue balls or purple or whatever on both sides. So again, equilibrium was reached. There is no net movement, right? And you can see again, those balls still are going back and forth, but at an equal rate. So there is no net movement of these molecules. Any questions about that side? All right, another thing that you probably already know because you've done pre-lab is this: factors that influence the rate of diffusion. And again, if you've noticed, and I did this on purpose when you did the pre-labs, there's the book definition, and there's the lab definition, there's the definition from other sources. So I'm not necessarily going to test you on this concept because which version would I test you on? But you do need to understand it to understand how diffusion works. So one thing is the concentration gradient. So again, using that, or not again, but if you imagine just a beaker and you put one drop of dye in there, imagine it diffused, right? But then imagine you had a beaker and you put a lot, right? The higher concentration of dye. You can imagine that's going to diffuse quicker because it's a higher concentration. The mass. Right? Generally speaking, the smaller things are, the quicker they move when we're talking about diffusion. Because, again, literally these are molecules running into each other. You might not think about that and see that when you see a beaker of water, for example, but they are. Those are molecules. Those are molecules that are in the way of other molecules. So if you were to put dye in there, again, it has to get through. Or more importantly, or a better example, this lab you're going to do today, you're going to have a petri dish, and it's going to have like a gel-like substance. And these things have to get through that gel-like substance. So obviously, the smaller they are, the easier they can get through that gel-like substance. So any questions about mass? All right. Another thing I would say too, that's, and I'm just throwing this out there to consider, for you to consider, that the book didn't mention, I don't think, and I don't think that the text does either. Excuse me, the lab. In my opinion, you shouldn't just consider mass too, but also the size. Right? So imagine you had something, two things that had the same mass. So I don't know, like 5 grams. Pretty big, what I'm describing here. But anyway, imagine one's like that big and one's that big, right? So they might be the same mass, but obviously something has a bigger size. Again, it has to get through all those molecules. So even size matters too. And by size, I don't just mean mass, like how much it weighs. I also mean like its surface area. Anyway, temperature. This should be very intuitive for you because we've talked about temperature so much. Hopefully this is, this makes a lot of sense. As you increase the temperature, what we mean on the molecular level is that molecules are moving faster. So obviously if molecules are moving faster, they're going to diffuse faster because that's all diffusion is. It's this movement of molecules. And then finally, solvent density. Right, which is basically like how thick is the stuff you're putting it in. So again, today's lab, you're going to have a petri dish that has this thick gel-like substance. That's going to be harder to move. You're also going to have a beaker with water in it, right? So water is less dense than this gel, so things should be able to move quicker throughout the water than they would through the gels. Now, granted, the experiments we're doing, those are two separate experiments, so we're not actually comparing gel to water, which would be a good demonstration, but we're not. Does anybody recognize or remember the one thing that's not on this list that was on the lab? There's one more thing. And again, for the exam, I'm not going to have it because there's just so many different answers. But all right, if no one wants to say anything, remember it's whether or not it has to cross a membrane. Okay? So obviously a membrane, at the very least, is going to slow diffusion down. Sometimes it just might stop it. Which brings me to some of the pre-lab questions. No one lost points for this. I really want you to get you thinking. For example, one person said that they believed that starch would diffuse out of the cell, like through the membrane and diffuse out. Then later they said they didn't think sucrose would go out. And not that I expect you guys to be experts yet, but if you're really thinking about what you learned in chapter 2 about micromolecules, sucrose is a disaccharide, right? It's just two glucose and a fructose. Starch it's a polysaccharide, right? So if you believe that starch could somehow get out of a cell membrane, logically, when talking about size and the fact that they're both carbohydrates, hopefully you would think that sucrose could also get out. But anyway, I got one off on a tangent there. Any questions about the factors that influence the rate of diffusion? Okay. Next word for temperature. Next word for temperature. The next word for attendance is mass. In this case, I'm talking about grams, like how much of something, as opposed to like midnight mass. All right, if there's no questions, we'll move forward. Download the PowerPoint. Watch this little animation that shows diffusion in action. So, substances that don't cross spontaneously are transported via proteins. So this is a conversation we had earlier when I showed you that picture that had two different proteins in that cell membrane. One of them was just like a hallway to let things through. The other one was like more of a mechanism that kind of took it in and spit it out. So we're going back to that concept. I said we were going to come back to it, and here we are again. That's what this picture is on the right? So some things, as I mentioned earlier, as this picture demonstrates with these little purple balls, they can just diffuse right through. Right? This is an area of high concentration. This is an area of low concentration. They are going to diffuse through. There's nothing, their size, their chemistry, whatever it is, they can make it through the membrane. These things cannot just make it through the membrane for whatever reason. Maybe the wrong size, maybe the wrong chemistry, who knows? The point is, they still have an area of high concentration to an area of low concentration, so they can still just diffuse, right? This doesn't require any energy. But then this one does require energy on the right. Again, you can see the mechanism, like the ball comes in and then it gets spit out the other side. Again, we'll come back to that. What I'm trying to teach you right now is something called facilitated diffusion. So facilitated diffusion is still passive transport, which is what we've been talking about. Diffusion is a type of passive transport. We call it passive, again, like I mentioned earlier, because it does not require an input of energy. And this is also the same. So facilitated diffusion is still diffusion. It is still passive transport. It still does not require an input of energy. The only thing is, again, the things just couldn't make it through the membrane, so we get a little help, like a little hallway. Sort of like imagine if we had, I don't know, 200 people in this room. And I turn the heat really high up, right? Everybody's gonna be like, I really want to get out of here. But the doors are locked from the outside and we can't get out, right? Air, we get a little bit of air coming in and out. You can consider that door a selectively permeable membrane because air molecules can get through the bottom, that crack of the door, right? Air molecules are going back and forth, no problem. But we, despite the fact that we want to diffuse out of this room, we can't, right? Because the door's there. So if you were to open the door, that would be facilitated diffusion in a sense. That doorway would become a protein that's letting you through. And then naturally, again, if you had 200 people in here with the heat cranked all the way up, people are naturally going to want to just leave the room. I wouldn't have to put forth any energy and say, all right, everybody get out, right? You guys would just want to do it, right? That's diffusion. That's facilitated diffusion. Any questions about this slide? All right, let's make it a little bit more complicated, but not much. Another type of diffusion, like I mentioned earlier, is osmosis. Osmosis is the diffusion of water across a selectively permeable membrane. So we're still talking about diffusion. It's just now it's really specific. It's water. And it's water crossing a selectively permeable membrane. So again, to remind you, what I mean by that is this membrane, it's like a cell membrane, it's letting some things in, not letting other things, but it does let water. So some terms that I mentioned, I think last week, and I think it was last week, I don't know. I mentioned them before, and I'm mentioning them again, and you need to know them. Solute, you need to know what a solute is. A solute is a substance that is dissolved in a solvent. So when you dissolve a solute in a solvent, what you're left with is a solution. I mentioned this when we talked about in chapter 2, we learned about water and the life-giving properties of water. And one of those life-giving properties was the fact that it's a universal solvent, was what the textbook called it. Meaning that it does a really good job of dissolving all kinds of different things. So that was your first time being exposed to these terms, but here they are again. Solute, solvent, solution. It'd be a little tricky because they all start with S. But hopefully, I don't know, even before I took biology, I always heard the term solvent, and I always pictured some liquid that dissolved stuff. So hopefully that, if you could just do that, then the rest of it should make sense. So yeah, again, you dissolve a solute in a solvent, and the result is a solution. Just for anybody who cannot see the screen, all we have here is just a couple of memes, so they're just for decoration. There's no reason for me to, I guess, talk about them. I will say this, though. I included this one. Yeah, never mind. It's all in there. But yeah, when people say, oh, you're learning through osmosis, no, because that would be water. You don't learn through osmosis because that would be water going into your brain. Anyway, any questions about this slide? Okay, if you download PowerPoint, you can watch osmosis in action. You can click on that video and it's animated. Here's where it gets a little bit trickier, and this is what I'm going to really slow it down here and make sure you understand before we move forward. Let's say oops, make sure we're still recording. Let's pretend that we're not talking about osmosis. Shoot, I almost kicked this off. Anyway, let's pretend that we're not talking about osmosis, right? In your brain, bring it back like 10 minutes ago when we were just talking about diffusion. If we're talking about diffusion, we're talking about well, it happens to be sugar molecules, whatever. We've got these green balls on the left, green balls on the right. Which side has a higher, well, it's already right there, right? Lower concentration on the left, higher concentration on the right. Actually, before I even move forward, I'm going to introduce you to these terms too. When something has a lot lower concentration, we call that hypotonic. When it has a higher concentration, we call it hypertonic. So hopefully you can just think about, like, what's it mean when someone says a kid is hyper? What do they mean by that? Hey, that kid's hyper. What does that mean to you guys? Especially some of you who are education majors and want to teach high school or elementary school. When someone says a kid is hyper, what do they say?
PhD.
Could be that. Yeah, like I, two of my children, or at least two have been diagnosed with ADHD. Yeah, and I might have too. I don't even know. Anyway, so yeah, that is part of it. What else? Like, even if a kid's not necessarily diagnosed with ADHD, and someone says like, "Hey, thanks for watching the kids. I had to go to the bathroom. How were they while I was gone?" And they say, "Oh, they were hyper." What does that mean? Hyper. High energy, right? They have excess energy. Thank you. We'll go with that. Excess energy. So just like this, right? When something's hypertonic, that has excess solute. This will be important. So you do need to be able to, you will have some questions where it's like, all right, is this hypertonic or hypotonic? Which is which? Anyway, that out of the way. Let's again pretend like we're talking about diffusion. Do those balls want to go from left to right? Or do they want to go from right to left? What do you guys think? Again, we're only talking about diffusion. Forget that we're talking about osmosis. We're not talking about osmosis right now. We're only talking about diffusion. Which way do those balls want to go? Right here. Yeah, to the left. Thank you. Because it's a higher concentration, right? These balls want to go that way. They want equilibrium to be reached. They want to have an equal concentration, right? We know that. That's what diffusion does. However, here we have the selectively permeable membrane, and it's not going to let those balls through. So if you were in charge, again, let's just assume this was just a whole, like a piece of glass. Talk about selectively permeable. Let's say it's not permeable at all. Like, it's not letting anything through. Let's imagine there was just a piece of glass right there. If I said, I gave you a glass of water or a thing of water, and I said, I want you to make these equal. Which side are you going to put the water in to make the concentration equal? The concentration of sugar. Because right now, again, this is really low concentration on the left, really high concentration on the right. And again, it is water, it is sugar, so you can almost think of it as drinking it. Like, you drink this, it's a little bit sweet. You drink that, it's too sweet. Right? That one's not sweet enough, that one's too sweet. So I give you some water and I say, fix it so they're equal. Which side are you putting the water in? The right side. Perfect. Thank you. Again, because look how concentrated that is. So since we can't move those balls around, like, I can't take the sugar molecule out of there and put it in here, but I can put some more liquid in there, and that's exactly what happens. That's what osmosis does. It does this naturally. Of course, in this case, it's not you pouring water into it. In this case, it actually does. It is a semi-permeable membrane, so the water does move. It moves that way until things are more equal. Now, granted, there's now more water and more solute on the right-hand side than there is on the left, but the concentration is equal, right? So any questions about this? Last term that you need to know then is, again, once it's equal, isotonic. Another I word for you. Isotope, remember, you'll need to have the exam from chapter 2. Isotope is when you have something that's the same element, but it has a different number of neutrons. So it's the same, but different. Right? Isomer is when you have something that has the same chemical formula, but a different structure. Isotonic, when you have the same concentration. So hopefully you're getting the gist here with these prefixes. Iso, similar, equal. Any questions about that slide? All right, so basically what we've described here is tonicity. This describes the amount of solute in a solution. That's a word I'm not going to use. I'm not going to use tonicity because I'm going to use the 3 parts that fall under tonicity, which is hypertonic, hypotonic, and isotonic. And you will need to understand that. You might get a picture on the exam, and it might look like this, where it's like, all right, you can see this on the bottom left right here. This is a cell on the right there. That colored part is the cell. You can see there's a lot of more solutes on that side than there are in this. So that means this cell is in a hypotonic solution. Right? It's surrounded by something that has fewer, a lower concentration of whatever that solute is. On the other hand, on this side, this cell is in a hypertonic solution, right? Because that surrounding the cell has a higher concentration than what's inside the cell. Meanwhile, it's equal right here in the middle, so we call that isotonic. The thing that you want to make sure you don't get confused of is what we're talking about. So again, if I'm talking about this cell and asking what kind of solution it's in, you would say, oh, the solution is hypotonic, right? Because the solution on the outside of the cell is lower. But at the same time, you can describe the same situation using different words. I could say the cell itself is hypertonic compared to the solution. Does that make sense? So make sure you understand, you pay attention when you get these questions on the exam. Pay attention to the wording. Because again, if I'm talking about this cell, and I'm describing the cell is hypertonic to the solution. The solution is hypotonic to the cell. Does that make sense? All right. Sort of like if my young brother were to come in here, some of you guys might consider me tall. But if he were to come in here, like if you're comparing me to one of you, you might be like, oh yeah, he's taller, she's shorter. And if you were to come in, you'd be like, oh, okay, well, he's taller or shorter, and the brother is taller. That's all relative. It's all what you're comparing it to. So any questions about this slide? All right. To survive in a hypotonic or a hypertonic environment, cells have to balance their water uptake or loss, right? Need to be able to control it, and that is called osmoregulation. Probably not going to ask you what that word is. It's just important to know in the back of your mind these things happen in your cell. It controls the water balance. And also, as you're going to see on the next slide, the words are here. It'll make more sense when I show you the picture. But if we're talking about animal cells, and I should have put that up there. If we're talking about animal cells, they prefer an isotonic environment. So that way things are, your cell's not shrinking or expanding in size. It's pretty much just staying the same size. Hypertonic's not good, and you'll see why it's make more sense when I show you the picture, because it dehydrates the cell. Hypotonic's not good because it might make the cell blow up. Again, I'm going to show you this on the next slide. So I'm not going to necessarily ask you about osmoregulation, but you might need to know this part right here. Well, I'll come back to that. So keep that in the back of your mind, and the next couple of slides we'll come back to this. This is talking about animal cells. Let's really quickly talk about plant cells. It's similar, but as you will learn once you watch the other video that I haven't posted yet, plant cells have cell walls, right? Animal cells don't have cell walls, plant cells do. Because of that, they have more structure and strength. So they can have this influx of water that's actually good for them. So what they like, and this again will make more sense here in the next couple of slides, but they like a hypotonic environment, which is what we have here, right? So because of the environment is hypotonic, what that means, again, there's different ways of describing this, right? This cell over here on the right, the solute on the inside, it's a higher concentration on the inside than it is on the outside. So because of that, water is rushing into the cell because it wants to equilibrium, right? It wants to equalize things. But for plant cells, that's what they need. That's what helps expand the cell walls and lets plants have the straight structure that they have. Of course, isotonic's not necessarily bad. Again, for animal cells, this is preferred. For plant cells, it's okay. They still have water coming in and out. But again, for them, they want that influx of water so they can push against the cell walls. But just like for animal cells, hypertonic is not good because, again, the water, if they're in a hypertonic environment, the water is going to leave the cell to go into that hypertonic environment to try to equalize things. And that's going to dehydrate the cell. Are there any questions about this slide? Okay, so as this mentions here, as the cells lose water, the cells shrivel, and the plasma membrane pulls away from the cell wall, and that usually kills it. So dehydration is bad, basically what we're saying. Probably not going to ask you anything about this. I'm just saying this is what happens, right? And this is a great example. I'm sure, I don't know if you've seen pizza at least. If you don't water them, they look like that. And I have a video somewhere where we forgot to water one in my house. And then I finally watered it after a long time. I took a video. It's not stop motion, what do you call that? Time-lapse video. So it was like a few hours condensed into a 30-second video. But it went from this to this in like 30, like I said, 30 seconds in the video. But yeah, so again, water is important for plants. Here's a nice little picture that's not from your textbook. I think this is from the old textbook. It's a great thing to study because it really compares and contrasts animal cells versus plant cells, whether we're talking about hypotonic, isotonic, or hypertonic, right? So again, for animal cells, when we have your isotonic, again, that's what we want, right? We want the same amount of water coming in that's going out. Nice and normal. For plant cells, that's okay, it works. Again, with the same amount of water coming in that's going out, they get flaccid, they wilt. Right? Now, with animal cells, again, in a hypotonic environment, that water is going to want to go into the cell because the cell is hypertonic to the environment. So the water wants to go in there to equalize things, but these cells don't have cell walls, so they just explode, right? We call that lysing. And then, of course, like I mentioned earlier, for plant cells, that's good. They have that incoming water, which lets them push against their cell walls, which gives them their structure. And of course, again, for both of them, if they're in a hypertonic environment, the water is going to leave the cell. And obviously that's not good. It'll dehydrate the cell, very often kill it. So are there any questions about this slide? All right. I think we can knock this out. Let's talk about active transport very quickly. By the end of this, you should understand how electrochemical gradients affect ions. I'm going to introduce you to that, and then you can brain dump it. Because again, I'm teaching out of the textbook, but for us, that's not important information. You'll be able to describe endocytosis, including some subcategories of endocytosis, which are called phagocytosis and pinocytosis, and receptor-mediated endocytosis, and probably just a bunch of words you're probably like, what's he saying? But again, this is what you're going to understand by the end of it, so I'm not expecting you to know it yet. And then finally, you should be able to understand the process of exocytosis, so let's jump into it. So remember, prior to this, we were talking about diffusion, which was passive transport. Now we're talking about the opposite of passive transport. We're talking about active transport. Active transport requires energy, as the name might suggest, right? We need this input of energy for this to happen. Using the same example I used earlier, if we had 200 people in this room and the heat was cranked up and there's like 5 people out in the hallway, it's going to probably take me some energy to get more people into the room to go against the concentration gradient. They don't want to be in here. It's hot, probably smelly. I'm actually going to physically push them in, right? They don't want that. So same thing here. Active transport requires energy. I might ask you this, probably not, but usually it requires ATP. ATP is going to be very important later in the semester. We're going to talk about it a lot, and I'll just say this. It's like we talk about ATP as if it's the only one, and as far as we're concerned academically, it is the only one. But there are some other things that provide energy. But it's, again, academically, we're only concerned with ATP. This requires a transport protein. So remember, the last thing we talked about was, or one of the last things we talked about was facilitated diffusion, right? So that was still regular passive transport. It was diffusion. It was things going from an area of higher concentration to an area of lower concentration. For some reason, they couldn't get through the membrane, so they had that protein that allowed them to just flow right through, right? But remember, then I showed you also in the same picture, I had that picture of the protein that would take the thing in and then spit it out. That is this. That is active transport. So it's not just sliding through the protein. We're requiring energy to make it change shape and spit that thing out, so to speak. Probably the most important thing to understand here with active transport is this bullet point right here. When we're talking about active transport, it's the opposite of diffusion in that things are going against the concentration gradient, right? It's like it goes against nature, so to speak, because it's not diffusing going from an area of higher concentration to lower concentration. It's doing the opposite, lower concentration to higher concentration. This concept will be important later in the semester. To truly understand how photosynthesis works and how respiration works, you need to understand this concept of diffusion and active transport and going against the concentration gradient. So that's important for a lot of reasons. The ones that I just mentioned, for example, photosynthesis and respiration, but also because it maintains a concentration of small solutes that differ from the environmental concentrations, which is another way of saying, all right, your cell needs something, right? It needs it. And the environment it's in doesn't have much of it. So nature is going to want that stuff to leave the cell, not come into it. But your cell needs it. So it's going to add, it's going to put forth little energy to bring that stuff into the cell, to import it, because it needs it. And it's not going to diffuse in because the the concentration gradient. Any questions about this slide? This one's completely unimportant. We're not going to talk about it again. It's in the textbook, so I'm just going to say it really quickly. This is probably the last slide of today's lecture. But the electrochemical gradient. The interior cell is negative relative to the exterior. I haven't even mentioned that, right? So we've just talked about concentration, the solute. But yeah, just so you know in the back of your mind, it also has to do a little bit with electricity. Yes, like I said right here, long story short, there are reasons other than concentration gradient that necessitate active transport that we don't even get into. The point is, we can't always just rely on diffusion. Sometimes we have to rely on active transport. Sometimes it's because of solute concentrations, and that's the one you need to know. Sometimes it's because of that. So last word for attendance will be long. Go ahead and send me those emails, a list of words, and then one question about the lecture. That's it. And then we'll be back here at 9 for lab. Matter of fact, if you want to send those emails later, you can, because we have a long lab today. So if you want to get up and stretch your legs or go grab a snack, whatever, you can go ahead and do that. We're also going to need to get all of our food and drinks to the hallway before we get started. I will say this about the long lab. A lot of it is waiting. So it's not a long active lab. It's not like you're going to be in there all the time constantly doing something. You're going to be doing something like once every 15 minutes, basically. So even though I am recommending you get your stretches out now or whatever, once we start the lab, you will be able to step out and come back in as needed. Heads up, I'm turning the lights on. Yeah, there we go. Right. Waiting on Logan with our cart of goodies. Haven't seen him in a while either, thanks to the weather. Yeah, well, there's like a unisex bathroom right here, the lock is really wonky. So also on the other end of the hallway is a, wait, no, I'm sorry, this is the bathroom on this side, sorry. Yeah, it's over here. It's been a while since I've used that one. Yeah. Yeah, I don't recommend going into that single-use bathroom. I don't know if you guys used it, that lock. Really weird. I don't know if you guys were here when my daughter was here for a lecture, but she got locked out and freaked out for a little bit.